Okay, so last week we did immunofluorescence. Welcome back to the channel. This week we will be doing fish fluorescence in situ hybridization. And you'll understand very swiftly why we started with immunofluorescence last week, because fish is just a variation of that, right? So recap of IF, you want to label some protein. So you make an antibody, or you buy an antibody that's already been made for that protein. That's your primary antibody, and then you use a conjugated secondary antibody that's conjugated with a fluorophore in order to be able to image the protein in question, right? And we went over briefly how antibodies are made, right? Well, this week we're going to be doing fish, which is very similar, and I'm just realizing that I stretched this image from this paper too far, so this is very blurry looking, whereas the actual image is not. We're just going to go with it. But anyway, it's worked. The protocol that we're going to go over today, after this very brief background, has worked for me in particular, and also this lab more generally, and several other labs for years. So it's a pretty reliable protocol, especially for, specifically for Drosophila. So the way that fish works, okay, very simple cliff notes here, and you can look at the channel. I believe there is a how fish probes work video that I did a while ago, but the basics are you're going to have an RNA probe that's going to have introduced some hapten, so like a DIG, a bio, a FITC, DNP, all sorts of different options, and that's going to be introduced on UTP. So you're going to have conjugated UTPs in your DNTP mix when you make your RNA probes. So then those proteins are going to be exposed for binding by your primary antibody. So it's just an IF. You're just doing an IF of RNA probes that are labeled with your antigen, right? So bio, FITC, whatever, right? DIG, right? So your probe is going to be the reverse complement of some RNA that you're wanting to label. You know, for fish, it's almost always going to be an mRNA or an intronic RNA. And then, so that's going to bind because it's complementary to the RNA in question. And then you're going to do your primary antibodies binding, and then secondary, and then you can image the fluorescence of your conjugated secondaries. Right? Straightforward. Very easy. So let's go right into the protocol real quick, and we'll go through each of the steps, what their purpose is, and that'll be it. Okay? Right. Okay. So let's just go through the protocol, and I'll explain each of the steps. So this is, like I said, a protocol that I know works for Drosophila, at least. If anybody expresses some interest, I have also a protocol that I've used in the past for zebrafish. I can go through that one as well, or I can just put it in the comments or something. If somebody wants that, just let me know. But it's basically the same thing. If I remember right, some clearing steps are different, but yeah. All right, so some recommendations here, just general recommendations, follow those. So step one, you're going to take the samples out that have been fixed, right? So a good fixation protocol is very, very key to getting a good in situ hybridization. And for this protocol, I am not actually showing the fixation steps or the fixation protocol because that is going to be, very different depending on species, of course, or cells. And, I can show you that in the future if you want. If somebody puts that in the comments, I'll definitely, or if somebody in the lab tells me that they want that, then I'll definitely put that up at some point. But it's a very straightforward process, right? You just fixing. Right, so you're going to have your fixed embryos. They're going to be kept at -20. And this is key to recognize. So the next steps are going to be washes, right? So you're washing in ethanol because in the -20, they're generally going to be in ethanol. Some fixation protocols might end with the embryos in methanol at -20, but they're probably already going to be in ethanol. So then you're going to do a couple washing steps, right, in ethanol, and then we're going to do a clearing step here in xylene. So it's actually 5 to 7.5 ethanol to xylene, and that's to actually make the embryos more transparent. And you're going to do this for 1 hour at room temperature, shaking. Yeah, so if I'm not sure, yeah, so unless it says otherwise in this protocol, it's always shaking. So this step is shaking. You're going to want to do this in the hood because xylene is super toxic. And you're also going to want to add the ethanol first, okay? And then you're going to want to add the xylene in 250 microliter increments. And the purpose of that is that the xylene is very corrosive. So if you add all 750 right away, or you add the 750 first, even worse, you're probably going to just dissolve all of your embryos. So you're going to want to put 250 in, invert the tubes a couple times, or put them on the rocker for a bit, and then add another 250, and then add another 250. Okay, and again, be careful with xylene because it's toxic. All right, so then you're going to want to do, again, some more quick washes with ethanol, okay? And these quick washes are going to have xylene, so make sure you deal with that appropriately. And then, more thorough washes with ethanol, and then back to methanol. I say back because in the fixation protocol, it's going to be methanol to ethanol. Ethanol is less corrosive, I guess. It's a little less harsh on the embryos, so it's better to store them in ethanol. But anyway, back to methanol, and then 50% methanol, 50% PBT. Okay, and again, PBT, all the recipes will be with this protocol in the description. So the purpose of this step is to begin rehydrating the embryos. You need them to be rehydrated in order to put the aqueous solution of your probes so that the probes can actually bind to the RNAs, right? So yeah, so add the methanol first, right? And then add PBT. Or you can premix the two and and then just add the 50-50 mix. But don't add the PBT first and then the methanol, because the embryos at this stage are very dehydrated, so they're hydrophobic. And so they'll actually make a little bubble around themselves, and then the methanol might not mix well to begin the rehydration process. Then you're going to do more PBT washes. And yeah, so all of the washes in this protocol are with PBT. And the T in this is tween 80, which again is a detergent. So again, a reminder that the purpose of the T is to permeabilize the membrane, right? Because it's a detergent, membrane is lipid. So you want to get deep tissue penetration, and you really want to get your probes into the cells, right? Okay. Next is going to be PBTF for 25 minutes at room temperature. So this is a refixation step. So you just did a rehydration, right? And you permeabilized the membranes. So you don't want to lose a bunch of RNA in your washes, right? So you're going to refix at this point. So this is going to be a formaldehyde refixation, basically. And that's, again, just going to better fix your tissues. So then you're going to do some washes to get rid of that formaldehyde. And then you're going to do a pro-K step, which is proteinase K. So you're dissolving proteins. So basically you're perforating the tissue to try to get maximal penetration of your probes into as many cells as possible in the embryos, right? And this is a very short step because you don't want to dissolve the embryos with the pro-K, right? And then after that, you're going to do a couple quick washes and then immediately refix again, right? And again, you're refixing because you just broke a bunch of stuff loose, and you don't want, all that loose stuff floating around, and you also don't want to lose any more mRNA, or whatever RNA you're trying to label. Most often mRNA. Then quick washes again, and at this stage it's important that you actually put your hybridization solution into the water bath so that it is warm. So before you start these washes, right? So you're going to have 30 to 40 minutes for that hybridization solution to heat up, and that's going to be in 55C. So then next you're going to be doing 500 microliters PBT, or 50-50. So PBT to hybridization solution, and you're going to incubate the cells at 55C for 10 minutes in that 50-50 solution. Okay. And then you're going to prepare for hybridization. So the next steps are just going to be a bunch of washes. You're just going to do a bunch of washes with hybridization solution. So every 30 minutes, you're going to change the hybridization solution and incubate at 55 for an hour and a half. I guess it's not a bunch, it's only 3. And then on that last wash, you're going to want to prepare during that last wash, you're going to want to prepare the RNA probes. And to prepare the RNA probes, you're going to need to denature your actual probe solution at ADC for 5 minutes. And the purpose of that is so that if they're all, hybridized together, you're going to break them up. It's basically a denaturing step like in PCR, right? And anytime you have your probes out, it's important to keep your probes on ice, right? However, don't do this step early and then put them on ice, because you're just going to rehybridize them to each other in the tube. So this should be done right before you're done with that last step, okay? This last prehybridization step. So this should be finishing right as you're finishing removing all of the hybridization solution from that last wash, okay? And then you're going to add 1 to 2 microliters. You're going to have to optimize for different probes, whatever the appropriate amount is, plus 100 microliters of the warm hybridization solution, and that's going to be your hybridization mix, right? And then you put your probes back as soon as possible, right? And then hybridization is going to be overnight at 55C, so at least 14, 16 hours, preferably 18, 20 hours, right? So then the next day, you're going to do, this is where the whole bunch of washes are. So you're going to use hybridization solution, and you're going to change the hybridization solution every 30 minutes for 2.5 hours. But in between each wash, you're going to invert it once, just to mix, to wash out as much of the probe as possible, because you don't want a bunch of background in your imaging later, right? And then you're going to get it back into PBT. So you're going to do a 50-50 wash with hybridization solution and PBT. And this is going to be at room temperature on the rocker, right? So, and then you're going to do a whole bunch of PBT washes, right? 1 hour's worth, 10 minutes each, so 6 of them. And then we're going to do our first antibody binding, right? So our primary antibody. So the primary antibody step is going to be blocking for 2 hours. So you can add blocking solution to the tube and rock it for 2 hours. And then you're going to remove as much of that as you can, and you're going to add 400 microliters of the blocking solution plus however much of the antibody is needed. For these purposes, it's generally 1 microliter. So, this says 1 microliter, but don't believe it, because if you're using some weird hapten, some rare one or something, and check the antibody, If it says 1 to 1,000, good. If it says 1 to 100, good. Just adapt and overcome. And then, treat your antibodies well, or you're going to have problems. And then, so this primary antibody step is going to be either overnight at 4 degrees C, or 2.5, 3 hours at room temperature. So that's going to be rocking. So if it's at 4 degrees, put a rocker in the fridge or the walk-in, and if it's at room temperature, of course, just put it back on the rocker. So that brings us to post primary antibody. So then again, you're just going to wash out the primary antibody. You don't want background. That's going to be a whole bunch of washes over an hour. and then you're just going to do the same thing for the secondary antibody, basically. So you're going to do a blocking step for 30 minutes, and then you're going to add your secondary antibody in blocking solution, and you're going to incubate 2 hours at room temperature. And it's important to remember that you're doing this step, this step and all future steps in the dark, right? So you want to wrap your tubes in aluminum foil or put them in a dark box, or do all of it in the dark room, whatever your preference, but needs to be done in the dark. You don't want to photo bleach your secondary antibodies, right? And then again, you're going to have to, for whatever whatever primary you're going to be using and whatever secondary you're going to be using, you're going to have to optimize and figure out the amount that you're going to need to use for consistent good results and images, and keep, obviously good records. And then after that's done, you're going to do some quick washes and then thoroughly wash out your secondary so you don't have more background. Then you're going to, for this protocol, it tends to work best with glycerol. I have tried per-mount as well. If you're going to use per-mount, I think it's actually better to do some kind of preparatory step. I can't remember, but in general, we just use glycerol for these. So we just then replace the PBT with 70% glycerol and let the embryos sink all the way to the bottom. Sometimes it's even better just to let them sink in the glycerol solution overnight, because they take a long time to sink sometimes. But you really want as little of this solution on your slide as possible when you go to mount. And then you're going to mount. And then that's it. And then you can just keep your mounted slides in the -20 for forever, basically. Just keep them dark, protected from light. And, they're going to be best within that first couple days to a week. And then after that, the image quality is going to get worse and worse and worse over time. But yeah, that is the 10,000 foot view of fluorescence in situ hybridization and this week's Tutorial Tuesday. So with that, I'll sign out and talk to you guys Thursday for some paper, but next Tuesday for Tutorial Tuesday. Bye-bye.